Good morning, everyone, and welcome to Epilepsy Research UK's Research Roundtable on Epilepsy and Genetics. My name is Quiva and I'm Head of Research at Epilepsy Research UK, and we have a fantastic lineup of speakers this afternoon who we'll be hearing from very shortly. But if we could just momentarily ask you to pause what you're doing. Today is, of course, Purple Day, one of the most important days in our community calendar, and a time when people across the world come together to raise epilepsy awareness. It's been a huge year for Pets on Zoom, so we thought, why not give them center stage this Purple Day? Whether it's a sweater for your setter, a cap for your cat, a cap for your cat, or a coat for your goat, why not dress up your beloved pets in purple today and help us to raise vital funds for research? If you could please share your photo on social media using the hashtags #PurpleDayPets, see what we did there, and Purple Day, and you could win a goodie bag later today. And we have had some absolutely brilliant photos submitted so far. Some of them are absolutely pawsome. Haha. So please do keep them coming, and you could win a goodie bag later today. That cat with the bow tie just makes me laugh every time I see it. Oops. Oh, excuse me, sorry. So back to the webinar. So epilepsy and genetics has been one of our most frequently requested webinar topics, and we have some brilliant speakers coming up. Firstly, we'll be hearing from Craig, who lives with epilepsy and suspects that there may be a genetic basis to his condition. He'll be talking about how epilepsy has interrupted his life, the challenges that he has had to overcome, and his hopes for future epilepsy research. We'll then hear from Dr. Charles Stewart, who works in the field of human genomics, which is the study of a person's entire DNA, and he's been working in that field for 27 years and also has a personal connection to epilepsy that he'll be sharing with us too. We'll then hear from Dr. Kate Baker, who is a clinical geneticist at the University of Cambridge and a member of Epilepsy Research UK's Scientific Advisory Committee. And Kate frequently works with families with genetic conditions, including epilepsy, and is often asked what the future holds for those affected. And then finally, we'll be hearing from Dr. Gabriele Lignani, who is an Epilepsy Research UK Emerging Leader Fellow at the Queen Square Institute of Neurology, University College London. Gabriele's research focuses on groundbreaking gene therapy, which may offer hope for treating epilepsy in future. Our speakers today have all contributed to our research blogs in March, so if you're interested in hearing more, you can visit our website, and the link will also be available in the chat box at the end of the webinar. Following the talks, we'll have a roundtable discussion session where our panelists will answer a selection of questions that have been submitted by you, the viewers, researchers, and our team. So firstly, we're going to go to our first speaker, Dr. Charles Stewart. Charles, are you able to share your screen, please?
Yes, of course. Just to say, I'm going to keep my camera off because in my rural location, my broadband is completely unreliable. So just to give my talk a chance of success, I'll keep my camera off. So share my screen. How does that look?
Brilliant.
Okie doke. Well, hello everybody, and it's an absolute pleasure to have been invited along to talk to you all. So I'm Dr. Charles Stewart, and I'm the Patient Advocacy and Engagement Lead at Congenica, a company that's situated on the Wellcome Genome campus near Cambridge in the UK. And I spent 27 years working on the human genome, and I actually was involved with analysing the human chromosome 10 back in 2004. I did my PhD at the Wellcome Sanger Institute as well, and I've also been involved in looking at genes associated with severe epilepsies. But more importantly, I'm the father of 2 children who have severe neurological disorders, and I'll mention them a little bit later. So the Human Genome Project was actually set up in 1990 to read all of the 3.1 billion letters that make up our genome. And the Wellcome Trust Sanger Institute, which as I mentioned, which is where I began my career, actually generated around 1/3 of it, which is the largest proportion of any institute in the world. And the Wellcome Genome Campus is also where my Congenica is based. And essentially, Congenica is a digital health company that provides software to help clinicians investigate the genomic basis of rare diseases. And we work very closely with Genomic England. So the Human Genome Project was finished in 2003, and it took around 13 years and about £2 billion to sequence the 3.1 billion letters that make up all of our DNA. The order of these bases, or more commonly called letters, is called our DNA sequence. And DNA is made up of 4 chemical molecules called nucleotide bases, and these are adenine, cytosine, guanine, and thymine. But they're often abbreviated to the letters A, C, G, and T, and this is how we consider them when we're looking at the sequence of the human genome. So traditionally, genomic investigations have looked at individual genes in isolation, sort of one by one, pretty much based upon the intuition of a clinician who might look at a type of seizure, for example, and think, well, that might be based upon this gene. But of course, if he gets it wrong, then it has to start all over again. And of course, this is very time consuming. More recently, a technique called whole exome sequencing has been used. And this looks at the sort of the 1 to 2% of the genome that actually contains the genes. So the rest of the genome, we're still beginning to learn as to what the sort of the remaining 98% does. Now, however, there are projects such as the 100,000 Genomes Project, and this is actually looking at the genetic basis of rare diseases and cancer. And they are beginning to investigate what the rest of the genome does. And we believe that this remaining proportion of the genome is actually probably involved in controlling genes, you know, how their genes are turned on, perhaps how genes are turned off. So humans are made up of trillions of cells, such as, for example, heart or liver and brain cells, and almost every single cell contains DNA, apart from things like mature red blood cells or perhaps the cornified cells in your skin, hair, and nails. And if you were to stretch out the DNA in each cell, it would be about 6 feet in length. That's 6 feet of DNA in each cell, and this is our genome. And we generally have 23 pairs of chromosomes, so one set from our mum and one from dad, although in some cases people can have an extra chromosome, such as in Down syndrome, which we call trisomy 21, where there is an extra chromosome 21. So karyotype is simply an individual's collection of chromosomes, and for simplicity here, I have shown just one pair. The chromosomes are numbered from 1 to 22 if they're what we call autosomes, and then we also have X and Y chromosomes, and these determine our sex. There's also the mitochondrial genome. You see right at the end of my karyotype figure, little MT. And mitochondria are found in cells and are responsible for energy production. Mutations in the mitochondria can be responsible for diseases because they have particularly large numbers of mitochondria in the brain. And neurological disorders, including epilepsy, occur quite commonly in mitochondrial disorders. So genomics is the process by which we read the letters in the human genome and then understand what these letters actually mean. Genes are responsible for producing proteins, and these are the building blocks for life and make us who we are and how we work. They're responsible for things like eye colour or our blood group, and even our ability to metabolize different drugs, which of course you can imagine is very important when thinking about which anti-epilepsy drugs to administer. And when things go wrong with genes, they can be responsible for disorders such as epilepsy. So this is my daughter, this is Imogen, and she was born at 29 weeks and spent 1 month on intensive care in 2012. And she developed fine until spring 2013. And she began to regress in her development. And then this happened. I don't know if you can see, her arms fly up into the air and her head rolls to one side. And then she makes a little cry, and then she goes back into these sort of curious arm movements. So we took this film to Accident Emergency, where she was diagnosed by this amazing man on the left here, Dr. Alistair Parker at Addenbrooke's Hospital, where she was diagnosed with something called West syndrome. And it's believed that the actual seizures that she's having there can cause the developmental regression that she was experiencing. But Dr. Parker, very clever man, knew about a clinical trial and put her on it, and that stopped her seizures within 24 hours. January 2015, and this is sort of around 2 years later, and Imogen's just over 2 years old. She's been diagnosed with cerebral palsy. And we spotted this. We have a night vision camera in her bedroom. And we went up to see what was going on.
That's correct, yes.
And this is me on the phone to the ambulance.
Yes, we're number 4 on the right. We're cream coloured.
As you can see, she's making sort of little eye movements and mouth movements and hand movements.
So
and what had happened is,
I can bring...
thankfully, our night vision camera had determined that she was having a seizure, and she was actually going through status epilepticus, which is essentially when you don't come out of a seizure. So anyway, she was then enrolled into the 100,000 Genomes Project to look for answers. So ultimately, the 100,000 Genomes Project was set up as a result of David Cameron's son Ivan's struggle with a type of seizure called Ohtahara syndrome. And we put her on there. You know, people often ask me why do I want a genetic diagnosis for my daughter. Well, it's to explain to her why she's different, family planning, more accurate knowledge of how her epilepsy might develop in the future, possible treatment avenues, essentially to help connect with others who have the same condition. So we got a negative result for my daughter on the day of my son's 12-week scan. It doesn't mean that her epilepsy is not genetic. Maybe it's just that the mutation for her is found in a region of the genome we don't know what it does yet. So anyway, this is my son, and he was born at 28 weeks. And sadly, he suffered catastrophic brain damage at birth. He has severe cerebral palsy, and he can't even stand and talk. And he was entered into a genome study at Addenbrooke's. Again, nothing was found, but I'm convinced that there is a genomic cause to both children's challenges. And I do believe that one day we will find a cause. Thank you, Quiva. So just some acknowledgements quickly. But thank you very much.
Thank you so much, Charles, for sharing your own research area and your family's work. I think it's so fascinating. It must be difficult for you having, working in the area and also having it affect your life as well.
Well, yeah, I suppose in some ways, perhaps it is all consuming, but you do take a lot of comfort out of actually, say, taking a bit of control back by working in the field, and you get to meet the most amazing clinicians and scientists, as well as, obviously, most importantly, other people who are going through similar tough times as us. So.
Of course. Well, we'll be coming back to you at the end when we bring all of our panelists back for the roundtable discussion. So we'll speak to you then, but thanks so much, Charles. We're going to try to share Craig's interview now. And so here is Craig, who we spoke with earlier this month.
Okay, so I'm here with Craig. Craig, thanks so much for joining us today. Before we jump into the questions that we prepared, I wonder if you could just tell us a little bit about yourself, where you're from, and what you're working as.
Yeah, so I'm Craig. I live in Norfolk, UK. I'm a master technician for Volvo Car UK. So busy doing that. I also run apprenticeships and so forth for motor vehicle technicians for the Volvo franchise. So yeah.
Fantastic, thanks so much. So yeah, we're going to talk a little bit about your condition today. You live with epilepsy, so I wanted to just jump in and ask, when were you first diagnosed, and what was your immediate reaction to the news?
You see, my reaction's tricky because I was actually first diagnosed when I was 4 years old. So to that extent, I didn't really have a reaction because I was too young to understand. The epilepsy continued until I was 14. So over time, it became a bit more daunting, a bit more controlling on your life, obviously, because I didn't have the opportunities that other kids did, because I had to obviously accommodate the epilepsy. So when I was younger, having the epilepsy, I used to carry medication with me that I had to take at the time. So that made it sort of difficult when you was out and about, because 9 times out of 10 you needed some form of chaperone with you to ensure it was all controlled and so forth. And the epilepsy when I was young was a lot more frequent than it is now. So it was difficult to keep on top of at the time.
Sure. So this webinar is about the genetics of epilepsy. You've mentioned that there's a possibility that your epilepsy could have an underlying genetic basis, and there are similarities between your grandfather's condition. He also had epilepsy. I wonder if you could talk a little bit about that and kind of what the genetic side of things means to you.
Yeah, so well, when I first started out with epilepsy, it was photosensitive epilepsy was my first trigger. But which my grandfather didn't have. But as I grew older, the epilepsy became more orientated by sleep. So sleep deprivation, stress, anxiety, things like that would trigger the epilepsy. Now that is exactly what my grandfather had. And at the age that it took over to be orientated around that, it was about the age my grandfather started. And he's continued through to, I believe he was about 42. So I'm hoping I follow that suit, where in a number of years' time I will come away from that. And the point of my grandfather having it was actually quite handy, because in a way you get an insight of expectation of what's coming, how to control it. So almost he'd gone through the phases of being able to learn to control it, and I could adapt to those routines and phases that he went through to help myself.
Well, that is interesting. Yeah, I can imagine it's kind of instructive as well. Yeah, that's interesting.
It gives you something to work to and work with to get the insight, and it did. It helped a lot, and it managed to control the seizures and live in a routine, especially around sleep, to learn to accommodate.
Absolutely, sleep's one of your bigger triggers, isn't it? If I remember rightly from your.
It is massively. Massively. So it's not a case of necessarily like late nights and stuff like that. It's quite tricky. But me and the doctor said to me that basically when I'm asleep, I'm not fully asleep, so I don't go into a deep sleep. So kind of your body's asleep, but your brain isn't, if you like. So I can have like 8 hours sleep and wake up extremely tired in the morning. Which was sort of mind-boggling for a start, because you would have thought that you would have been refreshed. So the sleep was causing it, and that's caused a lot by stresses, things like that, which have got worse over the years, obviously through careers and stuff like that. Obviously, the stress has piled on.
Next question here is, what would you say to anyone who's newly been diagnosed with epilepsy, or their families, as a result of your experience with the condition? I keep referring back to your blog that you wrote. I think there was some real good kind of handy tips there as well. So if you could talk about that, that'd be great.
First of all, the main thing, don't panic. Obviously, it's quite a daunting experience. You immediately think, oh my God, life's changing. And it is to an extent, but it's not as bad as you think it's going to be. The key thing that I cannot stress enough is routine. Routine is a massive thing. So whatever it is, I mean, in my case, being the sleep orientated epilepsy and so forth, it's the routine that you have of making sure that you follow the right rules that have been set for you, getting your sleep and so forth. Medications, make sure that you follow the rules of taking them. If it is your epilepsy, for instance, orientates around lights or photosensitivity, just set a routine so that you know how to control things and control your environment. And with that, it becomes a hell of a lot easier.
So thank you. So a few more questions here. We want to talk a little bit about the SHAPE network. So you might know recently we launched the SHAPE network to give people affected by epilepsy the chance to share their experiences and their research priorities, so we can work together to kind of focus more on the research that matters to people. And to get started with the SHAPE network, we have a few questions on our website. So I just wanted to kind of get your responses to a few of them, if that's okay now.
Yeah.
So the first one is, how has your life been interrupted by epilepsy? If there's probably countless ways, but yeah, the main ways in which it's been interrupted.
Yeah, I mean, the main key thing, I think it's difficult the younger you are, as in if you're a teenager and so forth, coming up. I say that the worst, time of having epilepsy is really between 14 and about 25. And your life is interrupted because the things you can do, the things that you can't do, and things like that. You also have a case of, for me, I get affected with things like memory and anxiety. These obviously have quite an implication on life, especially if you're trying to build a career and things like that. So it can have quite an effect on life that can be difficult.
At that age as well, 14 is difficult for anyone as well. So to add in the kind of condition,
I mean, the changes you're going through as a person at a young age and so forth also has quite an effect on it. Because the stresses on your body, the stresses on your life, exams and all that sort of thing. Me going up through the trade, working up to status of master technician is not easy, I can assure you. And the stresses and so forth really do contribute to the condition.
Sure. Next question here is, so what one thing that research could achieve would be life-changing for you?
Obviously, first of all, to be able to control epilepsy easier so that the seizures don't occur. But for me, that's not a massive problem as such, because the seizures are what they are. The thing that I struggle most with is the everyday life, the effects it has on the brain, the effects it has on your reactions, your memory, things like that. And I think if that were to be controlled a lot more and maybe helped in a lot of ways, I think that could make life a lot easier for somebody with epilepsy.
And finally, we're at the last question here. What are your hopes for the future of research? It's kind of comparable to the last one, I suppose. But yeah, what do you hope, say, in the next 5 to 10 years that research might be able to achieve?
I mean, over the last number of years, like since I was young and have it, the epilepsy treatments and so forth have come along a long way. But I think that is the main thing, as I said before, that, to try and improve the impact on life, I think would be the biggest thing that would be helpful to somebody with epilepsy.
Well, it's been a pleasure talking, Craig. Thank you so much for sharing your story.
Thanks for having me.
Yeah, and I would highly recommend anyone check out Craig's blog on our research blog on our website, where we'll share a link to that in a moment as well. But thanks so much for that, Craig.
Thank you.
So thank you so much to Craig for taking the time to speak with us earlier this month. I think he gave the responses that seizure control is one of his hopes for the future, but the impact of epilepsy on his everyday life. And that's something that we hear a lot from our SHAPE network respondents. So thank you so much, Craig. It was a really emotive story to share as well. And so now we're going to be hearing from Dr. Kate Baker from the University of Cambridge, who is also a member of our scientific advisory committee. So over to you, Kate.
Thanks, Kiva. Hello, good morning, everybody. I'm so pleased to be joining you on Purple Day today. I can't wait to see some more purple pets on Twitter later today. I don't have any pets, sadly, but I do have someone to share with you today. This is Bernadette, the brain. She's a 3D printed life-size brain model generated by an MRI brain scan collected at my research unit here in Cambridge. So Bernadette's joining us today. She's a bit chilly, so she's got a bit of a purple jumper on to keep Bernadette warm. I'll just put her over there so she can listen. So yeah, I'm Kate, and I work at Cambridge University Hospitals, also known as Adam Brooks Hospital, and I'm a doctor, a medical doctor in the clinical genetics department there. I'm also a research scientist at the MRC Cognition and Brain Sciences Unit, which is part of the University of Cambridge. I'm really lucky to work with the Scientific Advisory Committee of Epilepsy Research UK, where I get to interact with other doctors and scientists focused on epilepsy research. And we help with the SHAPE network increasingly to sort of spot the best future research opportunities that are going to make the biggest difference to people with epilepsy and their families going forward in the future. The research project that I run, which is not connected to ER UK or funded by ER UK at the moment, is called Bingo, Brain and Behaviour in Intellectual Disability of Genetic Origin. So if you're any Bingo participants or Bingo families, out there today. Thank you very much for all of your work with us. So I'm going to spend a few minutes trying to share with you my thoughts on the topic of, if genetics is the answer, what's the question? So here's epilepsy and genetics, the hope, or you might call it to some degree the hype. So the hope is that every person with epilepsy of unknown cause, which is the majority of people with epilepsy, would come into genetic medicine with information about their seizures, about their life history, about their health and development and their family, and that would be paired together with information about their genome to find their genetic diagnosis, so the specific cause of their epilepsy, and that we could achieve that for everybody. At the moment, we can achieve that for somewhere between maybe 1.25% of patients who come into genetic medicine. And a diagnosis would open this door to a brighter future in which we could really explain why that particular person has the epilepsy that they have, with their own seizure types and their own severity and their own triggers, and would explain why their epilepsy might be different from somebody with the same genetic diagnosis. We'd give clear recurrence information to other family members, and we'd give this information about prognosis, about what does the future hold for that person's epilepsy and the rest of their life. And it would translate to better treatments and improved outcomes, and this sunny future based upon a genetic diagnosis. So that's the hope. Can we actually achieve that at the moment? What's the current reality? Well, we can't achieve exactly that really for anybody, but increasingly we can do a pretty good job. Doing this better for more people means getting over quite a few hurdles. I'm going to use the hurdles because my little boy is a bit obsessed with watching the Olympics highlights on YouTube. He's just 3, and I've not been a very good parent during lockdown. I've let him watch far too much YouTube. Anyway, he really likes hurdles. So these hurdles are for my little boy. So the first hurdle is that not everybody with epilepsy of unknown cause is currently eligible for genetic testing within the NHS. For very good reasons, because we can't find a genetic cause for a large number of people with epilepsy. So the first hurdle is about inclusion. And currently we include individuals with very early onset epilepsy or epilepsy and additional features. Then there's a huge hurdle of equitable access for the population who are currently eligible. So I know that not everybody who could be accessing genetic medicine currently is, and we need to do better to increase equitable access. Then there's lots of hurdles about actually getting that genetic testing done. So there's a huge reorganization of genetic laboratory services and genomic medicine going on in the NHS, all in the middle of COVID too. So access to genetic testing in the last year has definitely not been what we want it to be. And we hope that this is going to improve over the next year or so. Then there are big hurdles with interpreting this information, putting together the genetic sequence data with the person's life history to be sure that we've got a correct diagnosis. Then there's the hurdle about actually explaining whether that genetic diagnosis actually matches to the person's own problems. So offering a really clear explanation of why that genetic diagnosis is the right one for that person. Then all the hurdles about the future information about family recurrence risk. We can say how likely it is that someone else in the family will have the same genetic sequence change, but within families that can translate to widely different risks of actual seizures and other outcomes. So we need an awful lot more information to provide really accurate recurrence risks for families. Information about the future and prognosis is sorely lacking, not just for improving what the seizure control is going to be like going forward, how seizures are going to change across someone's lifespan, but also how their general health may be affected by their genetic diagnosis. What progress with developmental skills and learning is a child going to have? What extra help might they need at school? Long-term, how vulnerable might this person's mental health be? And critically, can we use genetic diagnosis to help provide more accurate information about serious risks like SUDEP, like sudden unexpected death in epilepsy, like status epilepticus? We don't have good enough data on which diagnoses might translate to elevated risk. And in terms of treatments, our treatments are being increasingly guided by genetic diagnosis, but there's a long way to go there. So looking out into this bright, rosy future, it feels like there's a very big, dark, grey rain cloud over the current reality, such that you might not want to start on this journey in the first place. There is a way forward, and the way forward, I think, is a very hopeful one. So it's about removing these hurdles. And where are we going to begin? Well, I'm hoping that at kind of a policy and organizational level, we can start to remove the barriers between people with epilepsy who could be accessing genetic testing and those that currently do. We're going to improve our access to the best quality genetic tests that we have available through the reorganization of NHS genomic medicine services and more research. The more people who get into the system, the more genetic testing is done, the more data is available, the better we'll be able to interpret each person's genetic test result. But all these future hurdles about explaining why one genetic diagnosis can translate to such a widely variable type of epilepsy and widely varying outcomes for each person, that's a major challenge for future research. And that's, I suppose, where my research comes in. So we need more information that we can offer people after a genetic diagnosis so that we can really translate the genetic cause into genuine benefits for that person and their family. So do we need to wait for all of that information before we can work on treatments? Well, no, Gabriele is going to tell you about some really exciting avenues being explored for genetic information to inform new treatments. So that's going to happen and gradually fade away. We don't need to wait until all of the rest of the world is sorted out before we can work on treatments. So is that big dark grey rain cloud going to disappear altogether? Well, I hope it's going to be replaced by something a bit more like this, because genetics and epilepsy are complex. We're never going to be able to generate the perfect answer for each person. It's much more complicated than that and much more interesting. But my hope is that we can start to put the person at the heart of genetic medicine beyond the sequence. And then we'll be able to move forward down this quite complicated road together in a more hopeful way. I think I'm probably nearly up for my 7 minutes. I'm not sure. I was going to briefly mention my own research. So my research starts after genetic diagnoses. I gather together groups of people who share the same genetic diagnosis and find out more about what makes them similar and what makes them different and how they're different within the group, in their epilepsy, in their learning, in their mental health. Those are the things that I focus on. And then we try and get under the surface, bringing back Bernadette here, under the surface to say, okay, everybody's epilepsy and symptoms and learning and day-to-day life are different, but underneath the surface, how is that genetic information changing the way the brain has developed in its structure and its function to inform epilepsy risk and influence learning and social interactions and emotional regulation and all the other interesting stuff that Bernadette here does. So that's where I work. I also increasingly work with families. So although it's all about molecules and brain structure, we actually want to know how each person's genetic diagnosis changes the interactions between that person and their family members. So rather a wild idea to say that actually people with genetic epilepsies aren't just genes and brains in boxes, but they're people within families and social circles and society who actually need to understand the interactions between that person and the other people in their lives to better help everybody. So that's where I'm trying to move forward and remove some of those hurdles to making the most of genetic medicine. And I'm really keen to know in our chat today or in future events, what you, the people who this really matters for, think the questions should be going forward. Thanks for listening. And thank you to Epilepsy Research UK for bringing me into this fascinating community of people. And my funders and my co-workers are there.
Thanks so much, Kate. I loved the using the hurdles. I thought that was a brilliant way to illustrate. And do you think we'll ever get to a point where everyone undergoes genetic testing as standard as part of their diagnosis, or is that a long way off?
So it's coming in at an earlier stage of the diagnostic pathway for an increasing proportion of patients. So more people are getting tested to find a genetic cause at an earlier stage. Whether we'll get to a stage where it happens for everybody at the time of diagnosis is hard to say. I think it will no longer be, being rare is no longer rare. Looking for each individual's rare and unique cause of their underlying problems is becoming the expectation rather than the exception. One of the things I hope is that the many adults with epilepsy and associated problems who never had the opportunity to access currently available testing can be brought into the system, because it can still make a difference for somebody whose diagnosis of epilepsy was maybe decades ago. It can make a difference to their epilepsy management, their mental health, their understanding of their condition, and their family's experience. Plus, we as researchers and the new waves of children being diagnosed now can learn an awful lot from adults. So I think that we need to catch up with everybody who could benefit now, as well as helping new people coming through for the future.
Of course, and you've actually, we're not even at the question section, but you've answered a question that Judith kindly submitted about, can adults be tested to help learn for the future? And I think that's so important. We'll go on to Gabrielle's presentation now. Thank you so much, Kate, and we'll speak again in a moment. So now, Dr. Gabriele Lignani, am I pronouncing your surname right, Gabriele?
Yes, it's fine.
So we'll be hearing from Gabriele talking about his Epilepsy Research UK funded work on gene therapy and giving us an introduction to what gene therapy is in general. So over to you, Gabriele.
Can you see my presentation?
Yep.
Okay, thanks for the introduction, Kiva, and thanks Charles and Kate, because make my presentation easier, explaining all these concepts before. And I think that after my presentation will further underline why is important genetic testing and all what Kate just said. So I'm working on gene therapy for epilepsy, but before explaining what is gene therapy, I think that it's important to go back to what Charles said at the beginning, is what is a gene. So as all the language that they write in codes, also the biological system have the writing codes, that is the genetic code. And as Charles explained very well, there are 4 different molecules that you can call letters, that is A, T, C, G, that are bind together. AT is binding with a T and the C with the G, and then basically they define is the language of each cells in your body. And actually, as every language, you have your letters, but you have also your words, and your words are the genes. So every sequence of letters means something, right? That is called a gene. And the other important concept that each gene is producing something, the meaning of the gene, so the meaning of the word is called proteins, that are the most important, so are the components of your cells. So each cells express different kind of proteins, and which of these proteins, or which of your gene is active, define which cells you are and what you are doing, basically. So basically, if a cells in your body express only gene A and C, so only that 2 proteins, you can be a cells from your liver. Or if you express only B, you can be a neuron. This is a simplified, obviously, because if you think of how long is this code in each cells in your body, it's quite long. It's around 6 billion of these letters, around 2 meters long, or very compact in one of your cells. And you also have to think that you have 100 billion cells, for example, only in the brain, and neurons are more or less 100 billions. So you have a lot of genetic information in each of your neurons. And as I say, because it's a kind of language, if you write down all these letters of one single of your cells in a paper, like writing in a proper language, you will find that writing down all the letters in one single cells is more or less 1,000 times all the collection of Harry Potter. So it's quite long, the genetic code for just one cells. And in the human DNA, so in the human genome, you have more or less, as Charles said, 20,000 genes. So it means that you have 20,000 words that have a meaning, that are the proteins. And sometimes happens that you have some error in your genetic code. So sometimes just is a letter that is different, and then create a problem. So the meaning is different, the protein is not working, and you can have a neurological disease. Or sometimes you have an error in other genes that instead of not producing a proper protein, you can have a bad protein that is not properly functioning, and that leads to another neurological disease. So this is underlying why it's important to know the genetic information of the patients. So what is gene therapy? So gene therapy is an approach that leads to a modification of this genetic code. And it can be done in different ways. You can reintroduce an healthy gene, an healthy sequence to treat the disease. You can replace a sequence that is with mistakes with an healthy copy, or you can directly delete a gene that maybe is a bad functional gene, or you can do gene editing that I will talk about in 2 slides. That is actually correct the genes, the error in your gene. You can do in different ways. You can take cells from a patient, modify them and put them back, or you can use, for example, non-harmful viruses to deliver these tools directly in the cells in your body. So the classical way of gene therapy, is the basic classical way that gene therapy works, is if you have a mutation, for example, in a gene that leads to a non-functional protein, so you have an error in your genetic code, what you can do is to reintroduce in that gene. So you don't have it, you put it back, and you use, for example, adeno-associated virus that are not harmful and not pathogenic viruses that have these properties that they can enter in the cells and add something to your genome. And this is a bit the principle of also of the AstraZeneca vaccine. So they're using a different virus, but the principle is that they introduce the coronavirus spike protein, the gene that codified for the protein, and so the cell express that protein. Is the same basic concept. Another important concept with epilepsy, as you know, is that there is not only genetic mutation. There are several causes that leads to epilepsy. But if you think at the end, the underlying symptoms of all these etiology of these causes at the end is having epilepsy seizures. So another way to see this problem, instead of thinking of correcting the gene, is like trying to understand if we can correct the seizures in general, all the seizures, no matter what is the cause. And the analogy can be with the music. You have a distorted sound and you want a good sound. So the sound is, the music is a mix of different sounds. So what you use to trying to correct the distorted sound, you use a sound mixer that have different knobs that you can turn and you can change different properties and have a good sound. So for the brain activity, more or less is the same. You have epileptic activity. This is an epileptic seizure. You want to go back to a normal activity in your brain, so your cells your cells can go back to normal activity and function properly. So you can use gene therapy, introducing a protein. In this case, we, for example, introduce a potassium channels. That is a protein that makes the neurons be more quiet and calm down the neuronal activity. The last point is gene editing. And everybody heard about CRISPR. They got the Nobel Prize last year for this. And it's quite interesting tools. It's actually is a protein. So it's a product of a gene of bacteria, not humans. And has been engineered to take out from bacteria and using in human cells. So the principle of CRISPR is very fascinating because it's a tool, it's like a spaceship that can go around your genetic code and you can decide to use it to change your genetic code as you want. So basically, if you have a gene that codifies for a protein, you have a mutation that leads to a protein that is not anymore function, and you can correct it with the CRISPR and go back how it was before. So as I write here, imagine a world where we can basically correct your genome errors or your mutations, like you're doing using a Microsoft Word. And actually, you can use CRISPR in many different ways, also to regulate that knobs that I was saying of the sound mixer. So modifying how much expression you have of the gene, how much protein you want to modify the property of the neurons. So these open a lot of hope for the future, and obviously underline again how it's important to know the genetic background of the pathology to try to, for example, correct it. And thank you for the attention. Sorry that I'm 2 minutes probably over my time.
Thank you so much, Gabriele, and great to see that you're on board with Purple Day Pets as well. I think that's so interesting, that something that like gene therapy has uses beyond genetic epilepsies. I think that's huge, and just the whole concept of gene therapy just sounds like wizardry to me. We're going to be welcoming in Kate and Charles back now to answer some of the questions that people have been submitting to us. So here we are. Brilliant. Welcome back, both of you. And so I'm going to start with a question for all of you. The past decade has seen incredible advances in the field of genetics, as you've all spoken about during your presentations. And what do you think the next 10 years could hold, and what are some of the potential benefits for people living with epilepsy? Charles, would you like to answer that first?
Yes, of course. So I am hopeful that whole genome sequencing, so that is looking across all of our genome data, will become commonplace, a common and routine practice within healthcare. From a more scientific point of view, what I'm very interested in is so looking at how genes are turned on and off. And there's, as I probably mentioned in my talk, and there's a lot of the genome that we still don't understand, so-called, we call it sort of the dark genome sometimes, or just the bits that don't seem to have any function for protein coding genes. And I hope that we can begin to understand that, and that will help understand perhaps those people who don't yet have a diagnosis for their epilepsy. So that also means, what I'm hopeful for is that the diagnostic yield for genome sequencing will increase dramatically.
Wow. And how about you, Kate? What do you think the next 10 years could hold?
So I agree with Charles that in the next 10 years, testing will become the routine expectation, and that many more patients and families will receive a genetic diagnosis, and they'll receive it at an earlier stage in their journey through living with epilepsy. That's going to happen, but my hope is that we will be moving beyond the sequence, because what we need is to move beyond this new starting point of the genetic cause to having much better information about how that diagnosis relates to each person's own epilepsy, and how they can use that to improve their future. That's the longer term goal, but we're doing that already. So thinking, for example, one particular genetic disorder that I was involved in discovering actually in 2015, we worked very hard over several years to find the very first person with a new genetic diagnosis. But then within 18 months, I was able to be in touch with 10 people around the world with the same diagnosis. And now I know of more than 30. So that's still only 30, but that's over a couple of years. But my main job is really just gathering information about all those people, so we can put the genetic diagnosis in context and help new patients diagnosed with the same conditions. That's going to happen for all of the genetic diagnoses. Well, there are hundreds of them, so we've got a lot of work to do.
Of course, and that actually ties in with another question that we've had through on the chat, where someone has asked, rare genetic mutations, there might only be a couple of people in the UK, but there could be maybe 30 people across the world. And are there networks that scientists can use to link up people with the same gene mutation? Wow.
Yeah, there absolutely are. And they're really critical for finding the proof that a genetic variant really is a new diagnosis and really is a correct diagnosis for each person, rather than part of the background normal variation. One of the things that's confusing is that there's this fantastic momentum and global community all working together to solve, or trying to work together to solve these problems. there are quite a lot of patchy initiatives, and linking them up for the best for the benefit of each patient can be quite challenging. But that's one great position we're in terms of technology. Not only do we have genome sequencing, but we have global information sharing and an ability to gather information globally, and then translate that into new communities of patients and families who can provide each other with information and support, and can also drive the research agenda. So that's increasingly happening for rare genetic disorders. The global community of patients and families are driving forward our work towards better understanding their condition.
Of course, and hopefully this virtual workplace that we've all been working in for over a year now is contributing to better connectivity of people with rare epilepsies too. And Gabriele, how about you? What are your hopes for the next 10 years, given how much has been accomplished in the past decade in genetics research?
I have high hopes, and so I have to distinguish two different parts. The first one is for patients with unknown causes, and for acquired epilepsy, we are focusing on therapies to trying to treat the seizures without knowing the causes. And we have at least 3 clinical trials on pipeline in the next 2 years for that. But speaking about the genetic part, I think that it's really important what Kate said and Charles said, that we need to know the genetic information of the patients, because now we have the tools to correct and treat the genetic mutations, for example. But we need to know very early, because as Kate has also said, that maybe when we will know it is too late to treat the patients. So I think that the improvement of the genetic diagnosis is very important, because we can tailor the treatment early on, and probably we will have a big impact on these pathologies. But I think that in the next 10 years we'll change a lot of this, and we will have good progress, I think.
Of course, and I saw Kate had a hand up there.
I did. I just wanted to say again that so there's the global perspective that you can connect people together, but ultimately each person living with epilepsy is living in their home, in their town, in their street, in their village. And so one of the things I hope is that there'll be a general increase in understanding and awareness, not just of epilepsy, but of actually what underlies seizures. So that, for example, parents communicating with their child's school will find that in 10 years' time, their child's teachers or learning support assistants or medical professionals helping to look after them will have a better understanding of genetics and better able to use the information about the genetic disorder to help that individual person. And that it won't be so hard to try and explain to family members and friends about your child's epilepsy or their genetic condition, because we'll all have a better understanding. So I think it's important to bring it down to people's real everyday lives, as well as what happens kind of out in global media stuff.
Of course. And Charles, would you like to comment on that?
I mean, we have both my children, they attend nursery and school, and you know, we're very lucky with our school in the sense that they really accepted my daughter in, but there is still challenges in trying to get them to understand the challenges that we have every day. And I think unless you've actually lived through having epilepsy, and of course in Imogen's case cerebral palsy, it's very difficult for people to truly understand the challenges you know, that we experience. And maybe that's down to the fact you just cannot understand it unless you've been through it. But I think there's also partly to do with, you know, raising awareness. This is, you know, what we're doing today. This is on the webinar. You know, we're raising awareness of not just of the techniques that can be used to help people, but to actually understand, for people to understand the everyday problems that we have to deal with.
Can I come in again briefly, Kiva? I wanted to say that ways in which having a genetic diagnosis can build and help, and where I hope that Charles and his family do get there one day. Because I think my experience of working with people, when they obtain a genetic diagnosis, it gives you a new part of your identity, and a new way of getting under the surface of the symptoms of epilepsy to what's going on underneath. So something I wrote about in my blog was a very rare genetic disorder that can cause a similar kind of Rolandic epilepsy, in that we did a lot of studies showing that for that rare disorder, language and communication and speech and concentration were really big issues. So by recognizing the pattern that was linked to that genetic diagnosis and getting under the surface of the symptoms to the genes and the brain problem underneath, that's helped those individuals and their families advocate for their needs by saying, okay, I fit into this pattern because of my genetic diagnosis. So listen to me and listen to what I need. So it gives you a different template of what to expect for the future, and a different way of asking for help, and a different identity as well. So I think there can be an awful lot of those different benefits that can translate into everyday life after a genetic diagnosis.
Can say one very small thing. I think that also a network that I think that's something that here UK is doing very well, is trying to connect also researcher, clinician, geneticists, and patients, because I think that also a broader network with people involved in find therapies, understanding genetic cause, patients, families, and clinicians, I think that will help a lot to move forward all the field and trying to find treatment and understand better epilepsy as well.
And ultimately, my experience of doing genetic research in epilepsy has genuinely been that although there's a lot of technology and sequencing and amazing experiments, it really does start with listening to patients. So listening to patients in clinic who haven't got an underlying known cause, and trying to work out how can we solve that detective puzzle for this particular person and family. It starts with understanding their experience and listening to their problems. And in my research after genetic diagnosis, it genuinely does start with me traveling around, meeting people in their homes, having cups of tea pre-COVID, and just listening to people's life experience and starting to connect together what's shared amongst people with the same genetic diagnosis. So the more we can get those, more people who have been tested, get a diagnosis, the more we can start those new conversations that can start after a genetic diagnosis is found.
Brilliant. This has been the easiest Q&A ever. I've sat back and you've all continued.
Sorry.
No, it's fantastic. And yeah, it's so brilliant to hear such hopeful messages that you've all expressed, that the next 10 years, it seems like we're in for huge developments in this field. So thank you all. We've run a little bit over this afternoon, but I would just like to say a huge thank you to all of our speakers today, to Craig for sharing his experience of epilepsy, to Gabriele and Kate for sharing their research, and to Charles for sharing your research and your personal experiences of epilepsy and neurological conditions. It's been so fascinating, and thank you to everyone watching for submitting your questions. We've had so many questions this time and we haven't been able to get to all of them, so thank you so much for submitting them too. And of course, thanks to James and Becca, who have been behind the scenes, making sure that all of the technology runs smoothly, even when it doesn't work on my end. So thank you so much. And we also have a very exciting opportunity. You mentioned, our speakers today have mentioned the importance of people being involved in research and the role that people affected by epilepsy have to play. And so we of course last year launched our SHAPE Epilepsy Research Network. And today we also have an exciting opportunity for people affected by epilepsy to help shape the SHAPE network by joining our steering group and having a say in how the SHAPE network will run. So if you're interested in joining, please do get in touch by my email address that's on screen, and we'll be sharing a link to more information on the steering group in the chat shortly. Please keep those Purple Day pets, or Purple Day Bernadette the Brain, like Kate showed us. Please keep them coming in and helping us raise funds for epilepsy research. You've got until 4 p.m. to tag us in your photos and you could be in for winning a goodie bag. So we look forward to welcoming you to our next webinar, so ensure you are signed up to our newsletter to be amongst the first to hear. But a huge thank you again to all of our speakers and to you for joining us for this Purple Day webinar. So thank you all. Bye.